For effector T cells, there's CD8 and then CD4. That's nothing new, right? We know about cytotoxic T cells, and we know about helper T cells. But what we're about to go into really briefly in the next two videos is the specific ways in which all the subclasses of those work and the way that CD8 T cells work. And this is always because we're so selective in our processes here with the MHC type 1 and 2 compatibility issues. They have a really specific mode of action, and this always involves a direct cell-to-cell contact. So everything that your helper T cells and cytotoxic T cells, to an extent, work is by direct contact with cell, one cell at a time, which usually on the spot will result in the production and release of whatever type of cytokines we need to be making for that type of infection that we're dealing with. And certain types of effectors T cells are going to detach from the antigen-presenting cells, and most, the C here means cytotoxic, so cytotoxic T cells, and then helper T cells are going to leave the secondary lymphatics. The effector T cells express more adhesion molecules for the homing action, for interacting with infected cells. None of this is new material, right? We know about T cell homing and lymphocyte homing. We're just changing up the identities of certain types of adhesion molecules, in this context the vascular, which is selectively expressed on the inflamed endothelium, but also to increase the interaction with the infected cells. Notice we're talking about LFA1, CD2, that should be kind of remembering that. A lot of this is similar to the interactions that we saw with dendritic cells as well. CD45RO, or I've never heard of its name pronounced, so I don't know what the R and the O stands for, but understand that this is CD45 has that phosphatase domain that plays a role in activating a lot of the signaling cascades, and so this lowers the threshold response, meaning that they don't require the co-receptor. Remember, this is just for naive T cells, right? So once we're past that point in terms of development, and this is just a diagram showing all of the, in this context, it's the CD4 T cells, all of the cell surface molecules that are going to become activated upon activation, and then the ones that are present upon whenever they're at rest. So like whenever they're resting, they always have the L-selectin. They have LFA1, CD2, CD4, T cell receptor, obviously, and then the difference between the RA and the RO, again, is just with how much extent we have activation going on there. I'm not really thinking that this is that important. This is just identities of stuff that we've already talked about in terms of the mechanisms that they're involved with, so that shouldn't be anything superbly new. But yeah, changes in the integrins expressed in naive and effector T cells. So here we see the integrin LFA1 interacting with ICAM1, and then during the VLA4, and then the VCAM1, which is only present, to my experience, whenever we have some type of inflammation happening. And this is because the helpers T8 helper cell 1, T helper 2, and then T helper 17, as well as the cytotoxic T cells, these guys are all going to have to leave the lymph node and go to the actual site of infection to do their job. And activated endothelium VCAM1 is only present whenever the V, like vascular, on the endothelium is being inflamed. So you have, your anaphylatoxins, other types of cytokines, histamines, and things like that being released, just depending on the identity of the pathogen itself. And so the effector cells do not require the co-stimulation. We've already talked about that. One signal, that's the main point I want to say, is sufficient, right? We have done so much work in selecting for that MHC T cell receptor interaction thing that's going to happen, that's all that it takes for the T cells to do their jobs, right? One signal. It's not like with the innate immunity, especially like natural killer cells, those things depend on a lot of signals. This guy is just one. And then depending on what we're talking, if we're a helper T cell, we're going to be activating the target cell, or we're going to destroy it if we're a cytotoxic T cell. But the main point that I want you to stress is that it's one signal. I feel like I'm reiterating myself a lot here. Okay, so this is all stuff that I'm going to talk about in detail in my next couple of videos. I'm going to make a video for each and every one of these, I think. For the CD8 cytotoxic T cells, they have cytotoxins, and then they also produce cytokines, right? So we talked about CD8. We know what CD8 does. We've talked about this a million times from the very beginning of this course. We know what helper T cells do, and we have one Th1, for example, that deals with intracellular infections, Th2 that helps with basically mounts a coordinated attack to either kill a parasite or expel it from the body. We have T follicular helper T cells, which help with the B cells becoming activated and becoming plasma cells. Th17 This is going to help with extracellular infections. And then lastly, regulatory T cells, which kind of suppress the other actions of all these guys. The signaling for it, again, this is all review, Janus kinase. I've specifically made videos in my biochemistry stuff that really is kind of connecting to this. And I think I'll post like a little annotation to this explaining what the Janus kinase and all those other type of signaling mechanisms that we've kind of already talked about in the previous chapter.